The overall goal of this procedure is to quantify the muscle area in Drosophila adult muscles as a means to indirectly quantify muscle mass and characterize degeneration or muscle atrophy. For this purpose, we focus on dorsolongitudinal muscles because their size, distribution, and rostrocaudal disposition allow an easy orientation for transversal sectioning.
1. Fixation and resin embedding. The first step of the procedure is to anesthetize the flies with CO2 and, with the help of scissors and forceps, remove the legs, the wings, and the terminal part of the abdomen. Secondly, carefully transfer the carcasses, to avoid the deformation of the thorax, to a 1.5 milliliter tube on ice, placing a maximum of 6 individuals per tube, containing 200 microliters of solution 1. The carcasses cannot remain in this solution for more than 20 minutes. Finally, add 200 microliters of solution 2 and incubate on ice for 30 minutes. After incubation, remove all of the liquid, add 200 microliters of solution 2, and incubate the carcasses on ice for 1 to 2 hours. The next step is to remove the solution and dehydrate the samples through a graded ethanol series. Incubate the samples twice in propylene oxide for 10 minutes each incubation. Then, replace the propylene oxide with a mixture of